Pseudopodia. 35 languages. Amoeba protus extending lobos. Pseudopodia. This article is about eukaryotic cells. For the band, see pseudopod, band. For the podcast, see pseudopod, podcast. For the structure in insect anatomy, see prologue. This article needs additional citations for verification. Please help improve this article by adding citations to reliable sources in this article. Unsourced material may be challenged and removed. Find sources: Pseudopodia, News Times, Newspapers Times, Books Times, Scholar Times, JSTOR, October 2007. Learn how and when to remove this message. A pseudopod or pseudopodium, PL, pseudopods or pseudopodia, is a temporary arm-like projection of a eukaryotic cell membrane that is emerged in the direction of movement. Filled with cytoplasm, pseudopodia primarily consist of actin filaments and may also contain microtubules and intermediate filaments. 1, 2. Pseudopods are used for motility and ingestion. They are often found in amoebas. Different types of pseudopodia can be classified by their distinct appearances. 3. Lamellipodia are broad and thin. Phellopodia are slender, thread-like, and are supported largely by microfilaments. Lobopodia are bulbous and amoebic. Reticulopodia are complex structures bearing individual pseudopodia which form irregular nets. Axopodia are the phagocytosis type with long, thin pseudopods. Supported by complex microtubule arrays enveloped with cytoplasm, they respond rapidly to physical contact. 4. Generally, several pseudopodia arise from the surface of the body, polypodial, for example, amoeba protus, or a single pseudopod may form on the surface of the body, monopodial, such as Entomoeba histolytica. 5. Formation, edit. Cells which make pseudopods are generally referred to as amoeboids. 6. Frequently asked questions are the ways to give. I already donated. How often would you like to donate? One time give monthly. Please select an amount, USD. The average donation in the United States is around $13. $2.75, $10, $15, $25, $50, $75, $100 other. Maybe later. 1. Suzo RCN. 2. Next, choose payment method. Wikipedia still can't be sold. The 17th of October, an important update for readers in the United States. You deserve an explanation, so please don't skip this 1-minute read. We're sorry to interrupt, but it's Thursday, the 17th of October, and this message will be up for only a few hours. We ask you to react on the number of times you visited Wikipedia in the past year and if you're able to give $2.75 to the Wikimedia Foundation. If everyone reading this gave just $2.75, we'd hit our goal in a few hours. In the age of AI, access to variable facts is crucial. Wikipedia is at the heart of online information, powering everything from your personal searches to emerging AI technologies. Your gift strengthens the knowledge of today and tomorrow. Just 2% of our readers donate, so if you have given in the past and Wikipedia still provides you with $2.75 worth of knowledge, kindly donate today. If you are undecided, remember that any contribution helps, whether it's $2.75 or $25. Proud host of Wikipedia and its sister sites. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Via extracellular queue, edit. To move towards a target, the cell uses chemotaxis. It senses extracellular signaling molecules, chemotractants, for example CAMP4. Dictyostelium cells, 7. To extend pseudopodia at the membrane area facing the source of these molecules. The chemotractants bind to G-protein coupled receptors, which activate GT parses of the Rho family, for example CDC42, RAC, via G-proteins. Rho GT parses are able to activate WASP, which in turn activate ARP2/3 complex, which serve as nucleation sites for actin. Polymerization. 8. The actin polymers then push the membrane as they grow, forming the pseudopod. The pseudopodium can then adhere to a surface via its adhesion proteins, for example integrins, and then pull the cell's body forward via contraction of an actin myosin complex in the pseudopod. 9. 10. This type of locomotion is called amoeboid movement. Rho GT parses can also activate phosphatidylineacetyl 3 kinase, Pi3K, which recruit PIP3 to the membrane at the leading edge and detach the PIP3 degrading enzyme TN from the same area of the membrane. PIP3 then activate GT parses back via GEF stimulation. This serves as a feedback loop to amplify and maintain the presence of local GT parse at the leading edge. 8. Otherwise, pseudopodia cannot grow on other sides of the membrane than the leading edge because myosin filaments prevent them to extend. These myosin filaments are induced by cyclic GMP in D-discoidium or Rho kinase in neutrophils, for example. 8. Different physical parameters were shown to regulate the length and time scale of pseudopodia formation. For example, an increase in membrane tension inhibits actin assembly and protrusion formation. 11. It was demonstrated that the lowered negative surface charge on the inner surface of the plasma membrane generates protrusions via activation of the Ras/Pi3K/AKT/mTOR signaling pathway. 12. Without extracellular cue, edit. In the case there is no extracellular cue, all moving cells navigate in random directions, but they can keep the same direction for some time before turning. This feature allows cells to explore large areas for colonization or searching for a new extracellular cue. In dictyostelium cells, a pseudopodium can form either de novo as normal, or from an existing pseudopod, forming a Y-shaped pseudopodium. The Y-shaped pseudopods are used by dictyostelium to advance relatively straightforward by alternating between retraction of the left or right branch of the pseudopod. The de novo pseudopodia form at different sides than pre-existing ones. They are used by the cells to turn. Y-shaped pseudopods are more frequent than de novo ones, which explain the preference of the cell to keep moving to the same direction. This persistence is modulated by PLA2 and cGMP signaling pathways. 7. Functions, edit. The functions of pseudopodia include locomotion and ingestion. Pseudopodia are critical in sensing targets which can then be engulfed. The engulfing pseudopodia are called phagocytosis. Pseudopodia. A common example of this type of amoeboid cell is the macrophage. They are also essential to amoeboid-like locomotion. Human mesenchymal stem cells are a good example of this function. These migratory cells are responsible for in utero remodeling, for example, in the formation of the trilaminar germ disc during gastrulation. 13. Morphology, edit. Pseudopods can be classified into several varieties according to the number of projections, monopodia and polypodia, and according to their appearance. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The forms of Pseudopodia, from left. Polypodial and lobose, monopodial and lobose, filous, conical, reticulous, tapering. Actinopods, non-tapering actinopods. Some pseudopodial cells are able to use multiple types of pseudopodia depending on the situation. Most use a combination of lamellipodia and filopodia to migrate. 14. For example, metastatic cancer cells. 15. Human foreskin fibroblasts can either use lamellipodia or lobopodia-based migration in a 3D matrix depending on the matrix elasticity. 16. Lamellipodia, edit. Lamellipodia are broad and flat pseudopodia used in locomotion. 4. They are supported by microfilaments which form at the leading edge, creating a mesh-like internal network. 17. Filopodia, edit. Filopodia, or filous pseudopods, are slender and filiform with pointed ends, consisting mainly of ectoplasm. These formations are supported by microfilaments which, unlike the filaments of lamellipodia with their net-like actin, form loose bundles by cross-linking. This formation is partly due to bundling proteins such as fimbrins and fascins. 17, 18. Filopodia are observed in some animal cells, in part of Felicea, Rhizoria, in Testacea phylosea, in Vampyrlidae and Pseudosporida, Rhizoria, and in Nuclearitida. Opisthoconta. 4. Lobopodia, edit. Lobopodia, or lobose pseudopods, are bulbous, short, and blunt in form. 19. These finger-like, tubular pseudopodia contain both ectoplasm and endoplasm. They can be found in different kind of cells, notably in Lobosa and other Amoebozoa and in some Heterolobosia, excavator. High-pressure lobopodia can also be found in human fibroblasts traveling through a complex network of 3D matrix, for example, mammalian dermis, cell-derived matrix. Contrarily to other pseudopodia using the pressure exerted by actin polymerization on the membrane to extend, fibroblast lobopods use the nuclear piston mechanism consisting in pulling the nucleus via actomyosin contractility to push the cytoplasm that in turn push the membrane, leading to pseudopod formation. To occur, this lobopodia based fibroblast migration needs Nesprin 3, integrins, Rho-A, ROC and myosin 2. Otherwise, lobopods are often accompanied with small lateral blebs forming along the side of the cell, probably due to the high intracellular pressure during lobopodia formation, increasing the frequency of plasma membrane cortex rupture. 20, 16, 21. Reticulopodia, edit. Reticulopodia, or reticulous pseudopods, 22, are complex formations in which individual pseudopods are merged and form irregular nets. The primary function of reticulopodia, also known as mixopodia, is food ingestion, with locomotion a secondary function. Reticulopods are typical of foraminifera, Chlorarachnia, Gromia and Phylorata, Rhizoria. 4. Axopodia, edit. Pseudopodia, also known as Actinopodia, are narrow pseudopodia containing complex arrays of microtubules enveloped by cytoplasm. Pseudopodia are mostly responsible for phagocytosis by rapidly retracting in response to physical contact. These pseudopodia are primarily food-collecting structures, but also provide a means of hydrological transportation via the expansion of their surface areas. They are observed in Radiolaria and Heliozo. 4. References, edit. 1 Etienne Manaville S., 2004. Actin and microtubules in cell motility, which one is in control? Traffic. 5, 7 47077. Do. 10.1011/J.1000-1600-0854.2004.00196.x. PMID 1518084. S2 SID 230832215. 2 Tang DD, 2017. The roles and regulation of the actin. Cytoskeleton, intermediate filaments and microtubules in smooth muscle cell migration. Respiratory research. 18, 1. 54 do. 10.1186/12931-017-05444-7. PMC 5385055. PMID 283904525. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 3 Patterson, David J. Amoeba, protists which move and feed using Pseudopodia. tollweeb.org. Tree of Life Web Project. Retrieved 12 November 2017. 4 ABCDE Pseudopodia. Arcela.nl. 23 May 2017. Archived. From the original on 16 December 2018. Retrieved 16 December 2018. 5 Bajich, Burton J. Carter, Clint E. Eltman, Thomas N. 2013. General characteristics of the Euprotista, Protozo. Human parasitology. pp. 3751. Do. 10.1016/B978-0-12. 415915-0.00003-0. ISBN 978-0-12-415915-0. S2 SID 83272826. 6 Pseudopodia. Encyclopedia.com. Retrieved 16 December 2018. 7 AB Bosgrave L. and Van Hastert PJM, 2009. The ordered extension of Pseudopodia by amoeboid cells in the absence of external cues. PLOS 1. 4, 46266634. Bibcode, 2009 PLOSO, 4.5253b. Do. 10.1331/journal.pwn.0005253. PMC 2668753. PMID 193844419. 8 ABC Van Hastert PJM and Devriotes PN, 2004. Chemotaxis. Signaling the way forward. Nature Reviews Molecular Cell Biology. 5, 8626634 do. 10.1038 NRM1435. PMID 153666706. S2 SID 5687127. 9 Campbell E.J., 2017. A computational model of a moboid cell. Swimming. Physics of fluids. 29, 10 100 and 1902. Bibcode, 2017 PHFL. 29J1902C. Do. 10.1063/1.4990543. 10 Contima, 2008. Non-muscle myosin 2 moves in new directions. Journal of Cell Science. 121, pt 11118. Do. 10.1242/JCS.007112. PMID 180966887. S2 SID 163672236. 11 Hook, Andrew R. Chilkine, Alexandra, Mijian, Cecilo. Boltyansky, Rostislav, Dufresne, Eric R. Angenant, Sigurd B. Alschuler, Stephen J. Wu, Lani F. Wiener, Orion D. 2012-01. 20. Membrane tension maintains cell polarity by confining signals to the leading edge during neutrophil migration. Cell 148, 12175188 10.1016/J.cell.2011.10.050. ISSN 0092-8674. PMC 33087828. PMID 22265410. 12 Banerjee, Tatsat, Biswas, Debogiati, Pal, Dimansanka. Mayal, Yuchuan, Iglesias, Pablo A. Devriotes, Peter N. 2022. 10-06. Spatiotemporal dynamics of membrane surface charge. Regulates cell polarity and migration. Nature Cell Biology. 24. 10.1499 1515 do. 10.1038/S41556-022-00997-7. ISSN 1476-4679. PMC 10029748. PMID 36202973. S2 SID 2489909694. 13 Skoen Wolf, Gary, 2009. Lessons Human Embryology. 4th ed. Churchill Livingston Elsevier. 14 XUF, it al. 2010. Contribution of Pseudopodia to cell migration, a mechanical link between protrusion and contraction. International Journal of Cell Biology. 2010, 1. 13 do. 10.1155/2010/507821. PMC 29104778. PMID 2067197. 15 Mashiski LM, it al. 2008. Lamellipodia and Philopodia in Metastasis and Invasion. Feb's Letters. 582, 14 210211. Do. 10.1016/J.Febslet.2008.03.039. PMID 183961168. S2 SID 4643897. 16 carat AB Petri RJ, IT al. 2012. Non-polarized signaling reveals two distinct modes of 3D cell migration. Journal of Cell Biology 197, 3439455 10.1083/JCB.2012011124. PMC 3341168. PMID 225474008. 17 carat AB Bray, Dennis, 2001. Cell Movements, From Molecules to Motility 2nd edition. 18 carat Danijela Vinjevic, IT al. 2006. Role of fasciin in Phelopodial protrusion. Journal of Cell Biology 174, 6863875. Do 10.1083/JCB.2006030013. PMC 2064340. PMID 169666425. 19 carat Pseudopodium cytoplasm. Encyclopedia Britannica. Retrieved 16 December 2018. 20 carat Chengapa P. IT al. 2018. Chapter 7, Intracellular Pressure, a driver of cell morphology and movement. International Review of Cell and Molecular Biology. 337, 185. 211 do. 10.1016/bs.ercm.2017.12.005. PMID 29551161. 21 carat Petri RJ, IT al. 2017. Activating the nuclear piston. Mechanism of 3D migration in tumor cells. Journal of Cell Biology 216, 193 do. 10.1083/JCB.2016050097. PMC 5223602. PMID 2799890. 22 carat Reticulopodia. E-forums. Archived from the original on the 17th of July 2007. Retrieved the 30th of December 2005. Categories: actin-based structures, cell anatomy, cell movement. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.